Topic of today's session is types of fermenter. The course name is fermentation technology. Fermentation is a process of breakdown of complex organic materials by the action of microorganisms for the production of a commercially important product in a reactor. So there are several organic materials we are giving in the form of media, which contains carbon source, glucose, fructose, constiped liquor, and other protein sources, minerals, and all. That is the organic materials. The specific microorganisms we are introducing into the fermenter, that is the vessel, and the medium is converted into the product. The product may be the antibiotic or organic acid or vitamins, whatever it may be. So these processes are known as fermentation. The fermentation process is carried out in a bioreactor or otherwise called fermenter. A bioreactor is a vessel that provides a controlled condition which is suitable for the culture and growth of microorganism, particular species of microorganism. So there are different types of reactors. Here the 4 different types of reactor is given in your syllabus. The first one is CSTR, that is common stirred tank reactor. Second one is tower fermenter, and third one is cylindroconical fermenter, and the last one is airlift fermenter. So first we can see about the common stirred tank reactor and its component. So the stirred tank reactor, which is used in most of the fermentation process, it is an upright cylindrical vessel, and both the ends of the vessels are in semicircle basins. A stirrer is fitted in the central axis, and which is used for mixing of medium and air inside the fermenter vessel. Hence it is called as a stirred tank reactor. So it is fixed in the top side of the fermenter. So there are two lateral sides. These two lateral sides are it is having two opening. One is for inoculum, that is media is pumped into the fermenter, and the gas outlet is there in the top side of the fermenter vessel. Here at the bottom there are one opening, that is for harvest. After the fermentation process is over, the harvested medium is released into the harvested opening, and there is a inlet pipe for sterile air. Here the air is pumped into the fermenter and it is mixed with the stirrer. There are small narrow orifices are called the sporger. Sporger is a small narrow orifice. Through the orifice only the air is get into the medium and it is mixed by the stirrer. The air inlet is placed beneath the stirrer, and there is a heating coil to maintain the incubation temperature of the medium. This heating coil is used, and the total vessel, the fermenter, is covered by a water jacket. There is an inlet for the water jacket and the outlet for the water jacket. So if there is increase in the heat, then we can pass the cool water, and the fermenter can be, the temperature of the fermenter can be reduced. So heating coil is to maintain the temperature, and the gas outlet, the air is get into the fermenter, and after utilizing the microorganism, that carbon dioxide gas is released to the outlet pipe. Here there are two different sensors. The first one is a pH sensor, which the pH sensor which makes the medium pH that is sensitive to that microorganism. So to control this pH, there are acid and alkali pump which is fixed. If the pH is reduced, then the alkaline solution, that is NaOH or KOH, will be pumped into the fermenter and you neutralize the pH of the medium. So if there is an alkaline situation, then that acid HCl or H2SO4 is pumped into the fermenter. There is a foam sensor. By stirring operation, foams may be generated which can interrupt the air supply. Hence, the foam can be sensed by this foam sensor, and anti-foaming agents such as vegetable oils is pumped into the fermenter to control the foam. The second type of sensor is known as tower fermenter. The tower fermenter is non-mechanical agitator fermenter. Here, a long tube is placed in the fermenter. The ratio of the height and diameter of the fermenter is 6 is to 1 or 10 is to 1. So it's a long tube. Here, the bottom, there are sterile air is pumped into the fermenter. Through the movement or the air force, all the medium can be mixed. The microorganism and the medium can mixed thoroughly, and the microorganism can utilize the oxygen, and that exhaust gas will be released to the top side of the fermenter. Here there is an inoculum. Here the feed medium can be pumped into the fermenter, and here also some temperature may rise during the fermentation process. Hence the water jacket is present surround the fermenter vessel. So this fermenter is used for the production of citric acid, antibiotics such as tetracycline, beer and baker's yeast. So another type of fermenter is cylindroconical fermenter. The cylindroconical fermenter, it is having a cylindrical vessel and an inverted conical base at the bottom. So this fermenter is also having the water jacket for maintaining the medium temperature, and the inverted conical base which is having an inlet for carbon dioxide and a thermometer and outlet for harvested product, and one more cell harvest outlet is also there. Here we are culturing yeast, hence the yeast cell can be cultured as well as the product also can be harvested from the conical base. So the introduced carbon dioxide is utilized by the microorganism which is present in the fermenter. After the fermentation process is over, that exhaust carbon dioxide is released and it can be collected, and again it will be utilized into the carbon dioxide inlet. So the carbon dioxide can be recycled again and again. Here the thermometer is to determine the temperature which is present inside the vessel. So here this is the feed mixer at the top of the cylindrical vessel. The feed mixer can be introduced into the fermenter vessels. So the application of this fermenter is the production of beer and baker's yeast. So the next fermenter is airlift fermenter. This is also non-mechanically agitated fermenter. Here this fermenter is made up of two tubes. That outer column, the outer tube is made with the large mold-like cap, and the inner tube is made up of the open system which is fixed in position. So there are two vessels, that outer tube and inner tube. The outer tubes are known as downflow tube, and the inner tubes are known as upflow tube. Here the sterile air is pumped at the bottom of the fermenter. Through the action of or the force of air, the media is pumped into the inner tube. So this is known as upflow tube. So hence there are several spargers. Spargers are nothing but a small opening. Here there are several baffles. The metal baffles are present in the inner flow tube, which makes the medium forced towards the center. So the medium is get up and here then the medium, due to the force, it will be getting down. So likewise the medium is circulated in the upflow tube and downflow tube. So here there is a inoculum, there is a medium is pumped into the through this way, and so the utilized microorganism utilized this gas and it is released to the outside. So this is gas exhaust. So this is the harvested product is collected through this outlet for harvesting, and the outer tube is known as downflow tube and inner tube is known as upflow tube. So this is the mm, medium passed into the two tubes. Hence there is no need of agitator. So the microorganism can effectively utilize this uh, medium and produce the products. So the application of this fermenter is aerobic microorganisms being used in this fermenter. It is used for the production of single cell protein. To summarize these types of fermenter, there are 4 different types of fermenters covered. The first one is common stirred tank reactor, tower fermenter, cylindroconical fermenter, and airlift fermenter. So the structure and the parts of this fermenter is explained in this session, and the most importantly the applications of different types of fermenters are covered.